AP Biology, Chapter 22, Evolution, Part 8. Now that we have some information about the evidence for evolution, as well as some types of selection, we're going to review how inductive and deductive reasoning works with evolutionary theory. So how does this all fit in with the process of science? Remember, the process of science is inductive, taking the specific observations of nature, tying them together to form a general conclusion. We'll talk about how evolutionary theory was derived that way. Deductive is applying your theories and other things that you know to new situations. Science also relies on observations, experimentation, peer review, evidence-based approach to understanding how nature works. And using the process of science, we developed the sciences of astronomy, chemistry, physics, geology, and biology. Now we're focused on biology here. Some of the theories in biology are germ theory, cell theory, and evolutionary theory. Evolutionary theory, derived inductively, is an explanation of how things change over time in response to their environment. Now, how is the theory of evolution inductive? On page 5 in your notes, we took we had made a web of all the evidence for evolution, and then we went through it step by step. So using this information, we tied it all together, or Darwin tied some of this together, except for number 6, which was DNA evidence, and came up with the theory of evolution. So artificial selection, how did that influence Darwin? Well, if people can change things over time, Darwin reasoned that nature could also change things over time as a result of survival and reproductive benefits in the environment. The fossil record, how does that support the theory of evolution? Well, this is a chronicle of how life has changed over time. And the question is not if life changed over time, but how it's changed over time. And Darwin's mechanism of natural selection, resulting in the change in genetics over time, is the answer to why we have changes in the fossil record. Comparative anatomy, comparing homologous structures. Why do some organisms have similarities inside? Why do some have differences? Well, if we're talking about changes over time and adaptations to environments, organisms that are closely related are going to have more things in common because they have the same starting point as far as building these structures. Things that have more differences have a more distant relationship with the organism you're comparing it to because they have a different starting point, and their changes are more extreme as a result of more time and more mutations. However, comparative anatomy also explains things like vestigial structures, things like our appendix, as well as the impressions of the eyes on blind cavefish. In a Darwinian sense, they did not disappear as a result of disuse. They disappeared as a result of mutations that didn't have an effect on the survival or reproduction of the organism, and then were passed on, albeit in a non-functional form. Biogeography, that supports evolution because using the theory of evolution, we can figure out how things like marsupials evolved on places like Australia. Normally, placental mammals like squirrels and rabbits are better adapted to Australia, so why aren't there rabbits and animals that are placentals? Well, the answer is that they never evolved there, so they're not found there natively. Comparative embryology, we can compare the structures of different organisms like vertebrates, and we see that they have a common developmental beginning. Why would that be? The answer would be that if you're just modifying the structures during embryology to develop the structures in the fully formed members of the species, then you're still having to use the same starting point. You're still using the gill pouches and then modifying through mutations to produce the things like the lower jaw and inner ear. These things are explained through evolutionary theory. Then molecular biology, why would there be organisms that have more closely related as a result of more DNA sequences in common and less closely related as a result of less DNA sequences being in common? This is explained as a result of more time and mutations that results in more differences of DNA in isolated populations, and over time they become different species. How closely related you are to another species can be determined by how many DNA sequences that you have in common with those other species. And again, that supports the theory of evolution. And these things were tied together, all 6 pieces, tied together inductively to create the theory of evolution. So now that we have the evolutionary theory, what do we do with it? Well, let's talk about how we use the theories. Let's first of all talk about a little bit about experiments. Remember, science is testable. You have to be able to test your ideas by doing experimentations or predicting what you are expected to find in nature and seeing if your predictions turn out to be true. Also, experiments have to be falsifiable. If it's not falsifiable, if there isn't a possibility of getting results that don't support your hypothesis, it doesn't belong to science. Also, no number of experiments can prove anything in science. Proving stuff is the realm more of math, but you can disprove something if you have evidence that doesn't support your conclusions. However, in science, all we do is say we support our conclusions with more and more and more evidence. Even our laws and theories are the same way. We are pretty sure gravity is real. However, we never can prove gravity is real. All we can do is support it with more and more evidence. Scientists do not say they believe in evolution. Rather, they more accurately say that evolution is probably true based on observations of nature and testable experimentation. And you could say the same thing about anything in science, like electrons, gravity, heliocentric solar system, etc. So how do we use theories? Once you have a theory, the real value is that you can apply it to new things. Part of the atomic theory is that an element's identity is determined by the protons in the nucleus. A hypothesis based on the theory, so we're making a prediction here, if 3 protons are released from a lead atom's nucleus, we can change it to gold. And we have to have evidence supporting that hypothesis now. That would be the next step. And in a Russian lab, using high-energy radiation, the lead shielding became coated with microscopic small gold flakes. And we can actually turn lead into gold, except it's not very cost-effective. You make about $10 worth of gold from $1 million worth of process and extraction of the lead or the gold from the lead. So that's the atomic theory. Let's talk about the evolutionary theory. So this first step in using the evolutionary theory to form a hypothesis is select an adaptation you want to analyze. For example, an adaptation in elephants is that they're much larger than their predators. That's an adaptation, a survival benefit. The second step in using the evolutionary theory is ask yourself why this adaptation would be selected for survival and reproduction versus those that don't have that adaptation. And an answer for the elephants would be that elephants that were born larger were more likely to defend themselves from predator attacks, are able to survive and pass on their traits to the next generation. However, that's just a hypothesis. That's not evidence supporting it yet. So to actually support that, the next step is to support your hypothesis with evidence, fossil record changes, year-to-year observations of population phenotype changes, identifying a gene for a trait, etc. Fossil record shows a size increase in Asian elephants over time, so that supports that idea that they were growing larger were selected for larger size as a result of predation. Smaller elephants observed to be killed by lions more frequently than larger elephants. Again, that would be evidence supporting that idea that larger elephants have a survival advantage. All right, let's do another one. Another adaptation we're going to analyze here. Deer gather in herds. They have social behavior. When one is spooked, they all run, instinct for flight. So why would that be an adaptation? Why would that promote survival and reproduction? Herds of deer are more likely to spot danger than a single deer. You got more eyeballs to see danger coming. And also, if you have the instinct to flee when one is spooked, then you're more likely to survive and reproduce. If you're the one that stays behind when the rest of the deer get spooked, it could just be a branch falling or someone that's not going to hurt the deer, but it could also be a wolf or a mountain lion. And if it is a wolf or a mountain lion, you'll take your lackadaisical genes out of the gene pool if you don't get spooked with the rest of the deer. But the last step, you have to support your ideas with evidence, supporting your hypothesis with evidence. In this case, behavioral traits are difficult to get hard data for, especially to find the gene sequences of A's, T's, G's, and C's. However, evidence would be observing deer that don't travel in herds and comparing the survivability to support that statement. Typically in nature, what we do is we try to find a real-life example of something that already happens in nature, and that lends credibility to the hypothesis for a new situation. Sometimes I get questions about the human eye and how something like that could evolve. The answer is many small steps that also have survival advantages. So here we have complex squid eyes. We can just as easily use human eyes that have high visual acuity. Why would this be a survival advantage? Well, even detecting light from dark gives you a survival advantage, being able to hide from predators if you can detect the dark and move toward it. The direction of light and eventually even actually being able to see images are all survival advantages. So now we got to support this with evidence. What do we actually see in nature that shows a lot of these little intermediate steps or little steps that would lead to something complex like an eyeball? Well, the first thing that we see in things like some simple worms is an eye spot. It's just a pigment spot that can detect light-sensitive. They have light-sensitive cells that can detect just light from dark. You can't see any images. Even a patch of cells like this would allow you to see the dark versus the light and move away from that light if you're a prey animal, if you're a worm trying to hide from birds. Having a cup of cells, another mutation leading to a slightly incaved light-sensitive cells, gives you direction of light and dark. And if you can tell direction of light and dark, now you can more effectively move away from the light and hide in the dark if you're some kind of prey animal. We have the more complex eyes of the nautilus. Nautilus has almost a complete surrounding of those light-sensitive cells called the retina. Now we can form rudimentary images. However, they just appear as dark blobs. But even seeing a dark blob gives you a survival advantage if you can see a dark blob predator coming at you and moving away from it. More complex eyes, we have a glob of clear protein that acts as a refractive lens. Remember, mutations don't always have to provide beneficial adaptations. You probably had in the past of these creatures lenses that didn't work effectively. However, if you did have an ability to focus an image on the back of the retina, then that's going to produce an image that you can, if you're a predator, move toward, or if you're a prey animal, move away from if you recognize it as danger. Then, of course, the more modern eyes have muscle attachments to a lens that also have a complete circling of the retinal cells that detect light. And this is how an eyeball generally works. You focus an image on the back of the eyeball using the muscles that pull on the lens and form an image. Things like birds have even better acuity than us. They have a more packed layer of cells in the back of their eyeball that can detect even more sharper images. And this is the pathway of evolution that's thought to have occurred for complex eyes. Each step along the way provides a survival advantage. And if every step along the way provides a survival advantage, then it could be selected for by nature. And evolution could be the answer. So let's get some notes. So how do we use a theory? The first step, let's write this down. Make an observation of a survival or reproductive trait, an adaptation that you want to analyze. Second step, hypothesize how it was selected for. What was the survival or reproductive advantage? Third step, find evidence to support your hypothesis. For example, design an experiment, collect evidence of the same thing happening in other living things, analyze the fossil record for traits selected for over long periods. All those lend credibility to your hypothesis. So now we're going to look into some of the experiments that were done using the theory of evolution. Peppered moths. This is called industrial melanism. We see a change in the genetics of the population of moths over time as a response to light and dark environments caused by people. Here we have two different types of moths, moth phenotypes. As you can see on the dark background, the dark moths hide better. On the light background, the light moths hide better. In 1848, and we have data going back this far, so this is one of the first experiments that we see information for moth color going back over 100 years. The percent of dark moths were 5%, the percent of light moths were 95%. And at this time, it was mainly light-colored bark, and there was no soot or pollution in there because this was before the Industrial Revolution. 1895, during the Industrial Revolution, we had a lot of smog being kicked out of factories and covering the trees with soot, making the surfaces dark. And as a result, we see the percent dark being 98% and only 2% light. Now, they're not just changing because they need to change. Remember that we're talking natural selection here. The light ones stick out, they get eaten more frequently, leaving the dark ones behind to reproduce, and eventually the population reflects that trait, 98% dark. In the 1970s, we started cleaning up the environment in England. This is in England. And as a result, the tree color became lighter because there's less pollution on the tree. So the light ones now have an advantage. The environment changes once again. We have 81% light and only 19% dark. The dark ones are sticking out more and getting eaten more frequently. So during the early 1800s, pre-industrial England, we had low pollution, lichens growing on trees, light-colored bark, basically a place to hide if you were light-colored. During the late 1800s, industrial England, we had factories covering the trees with soot, killed the lichen, dark-colored bark. The dark ones had the advantage. In the mid-1900s, pollution controls, we had clean air laws, the return of lichen and light-colored bark, then the light ones had an advantage again. This is called industrial melanism. All right, let's get some notes.